Okay, so let's talk about the first part of chapter 3, and that's bacterial growth and culture. So this is going to be the theoretical components related to what's going on with bacterial growth and why we care about it. So first things first, when we grow in pure culture, we're using it to specifically characterize specific strains of bacteria, and we might also be using it to optimize commercial production of specific products. So we might want to be getting antibiotics or vinegar or specific things happening in pure culture. So pure culture is really about what we actually know about bacteria, whereas environmental matrices, or what we call in situ from Latin, is more complex than culture, and it's actually what's going on in the environment. So bacterial growth in pure culture is what we think of as in the lab. That's what we're going to be doing this semester. We're really looking at pure culture. We will use environmental samples, but we're using pure culture methods in labs. And it's really what's used for research. Doing things in the environment is very, very tricky because there are so many variables you can't control. There's a lot of confounding factors. So as a result, most of the time, pure culture is what we care about when we're looking at a specific strain of bacteria. So when we're growing in culture, that's what you're going to see on the right side of the screen. You're going to see these reactors that we make in the lab. This is, for example, growing algae. And you can see that we've grown one specific species of algae here, right? One specific item. Whereas the environment over here on the left, you can see there's all these other different things under the microscope. There's algae, there's fungi, there's all sorts of things. And many times we really can't dissociate what one organism is doing to the other or is doing to its environment from the environment itself when we're looking at environmental samples, which is what makes it so difficult in the scientific world to figure out exactly which microorganism is causing which specific effect. So as much as possible in the lab, we're over on the right side of the screen where we're looking at pure culture, whereas what is going to happen in the real world is what's on the left side of the screen. And we know how to grow microbes in the lab, but it doesn't tell us about their interactions to the environment. So one of the things that's very critical about how we're growing things this semester and how we grow things as environmental engineers to test for bacteria is that we are not capturing the interactions with the environment, which is the thing we do need to know as environmental engineers. So that does make the science very challenging for us as environmental engineers because we need to know the interactions in situ. So there's a lot of different ways we get around this in practice. Feel free to ask about it in class, but I just want to make sure that that's very, very clear going forward. So if we grow in environmental matrices, that's going to help us understand things like rates of nutrient cycling, the microbial response to climate change, microbial interactions with organic and metal contaminants, and survival and growth rates of pathogens in the environment. So if we have the ability to grow in environmental matrices, we try to, or we try and mimic them as closely as possible in the lab. So during the semester, you're going to see a UV reactor, you're going to see possibly a wetland that we've built, and these are meant to mimic an environmental matrix, and that's very different from growing in a petri dish. Growing in a petri dish is pure culture. And we can't understand how fast microbes cycle nitrogen and phosphorus. We don't understand what's going on with changes in temperature or contaminants. So many times we have to build what we call pilot scale reactors in the lab in order to mimic the environment. All right, so we've talked about metabolism before in class, and it's collectively those series of highly organized reactions in a cell. When we talk about bacterial growth, we start to break down metabolism in a different way than we've broken it down before. So we break it up into what we call growth metabolism and non-growth metabolism. So growth metabolism is the reactions in a cell that are utilized to make new cells. So that's reproduction, right? Make new cells in an organism. Non-growth reactions are needed for what we call cellular activities. So that might be the maintenance of intracellular metabolites, so other components involved in metabolic reactions. Maybe they need to repair cell structures. Maybe they need it for motility, so moving around. They need proteins or things like that, or in response to an environmental stress. So when we talk about bacterial growth, we're targeting the growth metabolic reactions when we culture things. We are not looking at the non-growth reactions. Those non-growth reactions are still happening, and we're going to see those later in the semester because it's related to cellular activity. And cellular activity is another way to figure out who or what's going on in a sample. But when we talk about growth in culture, we're talking about growth metabolism, and that's really important. So remember that there's two types of metabolism that we've talked about. There's anabolic and catabolic reactions. Anabolic reactions, right? Think anabolic steroids. It's going to be synthesis of cell constituents and metabolites. So it makes more bigger compounds, right? So somebody who's on anabolic steroids is the person who's getting very, very big in the gym and they're a bodybuilder, right? That's how you remember it's anabolic, creates bigger components. Catabolic is the opposite. It's the breakdown of constituents and metabolites. And most of the time we're going to look at anabolic reactions, but we can also use catabolic reactions to look at bacterial growth. That is a possibility. And the other thing I want to mention here is that when we compare lab and the environment, right? The environment, when we look at metabolic reactions, we're limited by environmental conditions, right? So whatever's in the environment is going to limit metabolic reactions. But when we're in the lab, we know the metabolic reaction that we care about. So we tailor our growth conditions specifically to that metabolic condition. And so we can actually get really, really quick cell division, quick as 10 minutes, right? You'll start to see as you count cells in culture this semester that we actually have given cells exactly what they need to do their metabolic growth reactions. So growth rates, right? What makes it faster? When we're nutrient rich, so we have nitrogen, phosphorus, and everything that a cell needs food-wise, and when we have what's called a homogeneous culture. So that means that we're trying to culture just one organism. So you saw this in one of our first labs where we went and we grabbed a sample of a very specific organism and tried to culture just that organism. And if it's homogeneous, they're going to grow better. Because if there's multiple organisms, which is called a heterogeneous culture, they will actually compete for the nutrients that are on a plate. So slower growth occurs when we have a nutrient-poor environment, so the agar is not optimized for the organism, or we have a heterogeneous culture where there's possibly competition between organisms. And what this photo is showing is it's showing binary fission. It's the division of two cells after replication of DNA. So growth in pure culture, here's the key things we need to know. We are going to provide the cells and the bacteria with all the nutrients they need. That's what agar is, right? Agar can be solid, liquid, or somewhere in between. We can make what are called batch cultures or continuous cultures. So a batch culture is like what we do in a petri dish when we have a finite amount of nutrients. You only have a specific amount of agar in a petri dish plate. But you can also do what's called continuous culture. So you can have a steady influx of growth medium and substrate so that the amount of substrate, what they're eating, their nutrients, are always staying the same. And then what we do is we count cells to determining mass, to figure out how much bacteria is there as a function of time. So which one do you think matters more to us as environmental engineers, batch or continuous? Take a pause in the video and just think about this for a moment. Which do you think? So for us, one of the things that we care about the most as environmental engineers is saving time and saving money because we are engineers and we are limited by economic factors. So in a batch process, if it takes us 10 minutes to grow, and then the next batch takes 10 minutes to grow, and then the next batch takes 10 minutes to grow, we've just spent 30 minutes processing a microbial culture. If we do continuous, we can grow 3 different types of bacteria all at once, potentially, in a smaller time, potentially. So continuous growth is actually going to be really important to us, especially in the wastewater industry, because if we are batching out a system, it's taking more time and it's taking more materials. So continuous is something we care about. In pure culture, sometimes you see these terms. So a consortium is a single organism or a group of organisms that are growing. Substrate is the food that organisms need to grow, and media is what you're growing the organisms in. So substrates can be considered food, it can be considered nutrients, and then the medium is the agar that we're growing them in. So I just want you to be aware of these terms because different textbooks and different papers use slightly different terms. These are the technical biological terms that we use for pure culture growth. And then in growth in pure culture, we measure growth by counting cells or determining mass as a function of time. So what you're going to see on this particular graph, and we're going to discuss each of these phases in the next video, is you're going to see time increasing, and you're going to see what we call log base 10 CFU per milliliter. So we count colony forming units, but we do it on a log scale because bacteria are exponential when they grow. And there are defined phases. So we measure cell counts or we figure out mass. So the way that we can do that sometimes is we can either just count on a plate, but sometimes that doesn't help us because we might be growing continuously. So we can actually use what's called optical density to measure how much light is able to go through the media. The more light that goes through the media, the less bacteria there are. The less light that goes through the media, the more bacteria there are. So it's an inverse relationship, and basically the bacteria are blocking light from going through. So characterizing bacteria using optical density is what's shown in the red line on the graph. It's showing you basically the density of cells over time. So that's one way we can do this besides just measuring counts on a plate. If we count on a plate, we would do dilutions just like we always do, and then we would back calculate using the CFU calculation that we've learned before. So that's going to be the way that happens. So it's exactly as we've seen in the past. We tend to use that log base 10 again because we have so many bacteria in our cultures. And then lastly, which one do you think is more cost effective, counting cells or optical density? Take a pause, think about it for a moment. So it's actually going to be optical density. You saw how much time it took in our very first lab to count cells and create plates and do all of that. That's very time consuming. It's very material heavy. So optical density, and this would be something that we call a proxy measurement or a surrogate measurement, is going to be the most effective for us. That's going to round us out in terms of theory. In the next video, we'll talk about the different growth phases.